All right, welcome back. This is chapter 12. It's kind of a short lecture because we're just going to focus on just cell cycle and mitosis. So when we talk about cell cycle, it is the process that cells go through during their lifetime. Some will go through this process and other cells will not. All right, so we're going to go through each of these steps. All right. So the cell spends most of its time in interphase. Interphase is from here to here. So it includes growth one, S phase. So you can see here G1, S, and G2. All right. A lot of cells actually go out of the cell cycle into what's called, oh my gosh, this is awful, called G naught. All right. We call that it's arrested and it never may go through. It may never make a copy of itself. So again, that's referred to as G naught. This should be like um, a superscript zero. So again, it consists of G1, S, and G2. All right. So G used to stand for gap because we weren't sure what was going on during that part of the cell cycle, but now with advanced technology and better equipment, we know that at least in both G1 they're growing because when it first gets done reproducing, the cells are about half the size. So it's going to spend time growing um, and then doing what it's supposed to do, its regular job or function during G1. All right, G2 over here, if we have G2, that again, the cell's going to get bigger in order to split into two cells and prepare for mitosis. We also have to increase our number of organelles because we need mitochondria for two cells and and Golgi apparatus for two cells and smooth and rough ERs, all those things. In synthesis, what's going to happen is we're going to go from our unduplicated to our duplicated um, chromosome. And this is often called monad for one or dyad for two, the two sisters. You have what's called sister chromatids. They're identical. And we're going to get into how exactly DNA copies itself. But during synthesis, that is where we go from one copy to two because we need two copies, one for each cell. And then gap two, again, we're going to make sure that everything's ready to go. We have the microfilaments and all the different types of structures, cellular structures we need in order for the cell to divide. Now, the meiotic phase, and this is the key, the meiotic phase is nuclear division. This is where we're going to divide the contents of the nucleus, hence the DNA. Then we have cytokinesis, and cytokinesis is where we're splitting the cytoplasm. Remember, in the cytoplasm, you have the liquid cytosol and all of the organelles. Wow, that would be sad to have a heart inside of a cell. So our organelles. Control of the cell cycle, we have what are called growth factors. These are external signals that will tell the cell to finish its cycle. It can, again, also give those signals that nope, you're going into G naught, you're not going to be divided. Generally, we divide cells like when we have an injury, right? We need to repair those cells. Or if we're growing still, right? If when you get taller, of course, once you reach maximum height, you might grow horizontally versus vertically. All right. So, and we do a lot of research on stem cells, and that's just where we're looking at because there are certain cells, like specifically nerve cells, that will never divide. And so, when you have some kind of spinal cord injury, we're looking at trying to take some of those stem cells and regrow those nerves. So, it's a big area that we're working on is regenerating nerve cells. With the cell cycle, we have checkpoints. We have the first checkpoint, again, is in gap one. And again, it will tell it whether to continue dividing, keep going around and around in that cell cycle, or to go into G naught. Like I said, most of our cells are in G naught unless we need it. There is a protein that helps there's actually a couple, there's P53 in our cyclins. Not going to get too much into that, but the protein P53 will stop the cycle when there's damage detected in the DNA. When that damage is detected, if it can't be fixed, the cell will undergo kind of like self-destruct with a process called apoptosis. And when apoptosis occurs, that's called programmed cell death. It will round up that cell. You can see we got the bad cell in there. It'll kind of round up and then what we call bleb, which means we've got all those little pieces, right? So those blebbing and blisters, and then the cell is broken down. G2, we have a checkpoint. Make sure that the DNA is duplicated with no damage. If damage is detected, it is repaired. Again, if it can't be fixed, we undergo apoptosis. M. During mitosis in metaphase, you're going to have another, actually, your last checkpoint. You're going to check that your spindle fiber assembly is all hooked up to the chromosomes and everything is good to go. We have the right number of chromosomes. And if not, again, cell death if we can't fix it. So here's an example of, somatic cells are body cells. And when you're undergoing development as a fetus, there are some tissues, especially between your fingers and toes, there are skin. Your hand kind of starts off like a paddle. And so you have to, right? This is where we have properly caused cell death in between and killed the skin between the toes. As you can see here, didn't work all the way. All right, so the message did not get relayed to those cells to undergo apoptosis, and this person has some webbed toes. You can have webbed fingers, webbed toes, and you might not think that that's a horrible thing to have, but it is a bad thing if the cell is cancer, right? So if your cell becomes a cancer cell and you cannot detect that it is not made properly, this is what leads to cancer. So chromosomes, we see how we organize them. We're going to wrap them all up. Actually, sorry, I got ahead of myself. This is the double helix. And then we have these proteins here called histones, and we're going to start to wrap our DNA around the histones. And then we're going to tighten those up. Notice how we have all this open space here. We're going to close that space up, wind it up even tighter. And then we're going to end up starting to coil up those areas. And then we have what's called supercoiled. And then eventually it gets us to this structure here, right? That's the chromosome that we always see represented as an X. So, we represent it in this little cartoon when you see the DNA is like wound 300 different ways. And you can see the electron microscope picture here showing you what that looks like. So each chromosome, these are identical. These are called chromatids. The whole thing is a chromosome. The arms or the one side here of each those are identical sister chromatids. They're going to pension here at the centromere. Okay. Then we have a protein complex that sits out here in this pinched in area called the kinetochore. And this is where your spindle fibers are going to hook up to the chromosome in order to divide, put what we're going to send one copy to one cell and the other copy to another cell. Now, kind of where all this master controls are coming from is in the centrosome. And the centrosome is made up of two centrioles. You see them here. And then we have the spindle fibers coming off of those centrioles. We talk about the centrioles having this 9+0 triplet organization. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. And we don't have any in the middle, which is where the 0 comes from. So this is how those are organized there. So when we talk about the stages of mitosis, we're going to start with early prophase, and during that time, the nucleoli will disappear. The chromatin, we're going to wind up our chromatin into nice, tightly wound chromosomes. The centrioles are going to move to the opposite ends of the cell. And the nuclear membrane is going to start to break up. Here's a picture under the microscope. And if this was interphase, again, the nucleus would, and I'm putting it red because we do a lot of stains with that red, will kind of have a shaded in nucleus versus where we have these little blotches where we're finding. All right, so again, this is more like we're getting closer to late prophase. Prophase takes a long time. So we could say early, late prophase. Now we have a prometaphase, which is kind of in between. So here we can see the chromosomes are really winding up now. And then this would be kind of more towards like the middle of prophase here or late prophase. This would be going towards prometaphase where our chromosomes are actually hooked up and getting prepared to move into metaphase. So here's our prometaphase. You can see they're all kind of in the center here. They're getting ready. They're getting attached. We're playing a little tug of war. Again, with the spindles hooked up to the kinetochore. Now, here's that kinetochore, right? So we have that protein. You can see the spindles coming out. And then remember, the pinched in area here is referred to as your centromere. And there's another picture. So when we get to metaphase, I always remember middle. Metaphase is middle. They're going to be lined up in the equatorial plate. That's the middle of the cell. So that would be our equatorial plate, or it's also called the metaphase plate because this is what we're seeing, um, the middle in metaphase. So they're all going to line up down the center. We've got our centrioles here. We have our spindles coming off and attaching to the kinetochore. This is a very short phase as it basically lines up and then splits into, here's a picture of that under the microscope. See how they're all lined up. And then anaphase, I always think of like apart. It's when we're moving those sisters to the opposite poles. And you see how we're splitting and we're moving them to the opposite sides of the cell. We split apart that paper clip, that centromere that held those, in the kinetochore that held those two sister copies together. And here's a picture. You see the chromosomes moving apart from one another. And then we have telophase. I always think of the end. And it is. It's the end of mitosis. It's not the end of cell division, but it is the end of mitosis. The cell's going to elongate. You can see the chromatids are reaching their maximum distance apart from one another. They do start to become less distinct because will start unwinding. And then we have these daughter cells. That's what we're going to call each one of these. These are two daughter cells. And then they're going to undergo what's called cytokinesis. To me, I always think this looks like googly eyes. Okay, so you can see here, you can see where we're pinching in here. Cytokinesis, that is again, we talked about a little bit in the beginning, that's the division of the cytoplasm. The organelles that doubled in size are going to divide by binary fission. We talked about endosymbiotic theory, especially if we were to look at the mitochondria. They will reproduce by binary fission because they were at one point their own independent prokaryotic cells. So with an animal cell, we can pinch it. And this where we're pinching is called the cleavage furrow. And we're going to have actin microfilaments at the equator of the cell. And I always think it's kind of like a pull string purse, and you're just going to kind of pull it closed. And then eventually the two cells, ooh, trying to go back. Oh boy, I'm trying to go back, guys. Sorry. It's not letting me. Eventually that, like I said, those purse strings will pull those two cells apart. Cytokinesis in plants is a little bit different. Oh my goodness. Let's just do it that way. Okay. So remember that plants have cell walls, so we can't pinch them in like we would an animal cell. They also don't have any centrioles or what we call asters, and asters is, kind of like it looks like an asterisk. It's that centriole with um the spinofibers coming out of it. That cell plate forms at the equator. Okay, that is actually you're going to have cell wall materials that's going to be secreted by the Golgi apparatus. It's going to send those vesicles that will start to um, accumulate at the center and put in your cell wall there in that structure. So that structure dividing, starting to be formed is called the cell plate until eventually we will have those two cells completely divided into two new daughter cells. Okay. Mm. And like I said, this chapter was going to be short and sweet, to the point. If you have any questions about the cell cycle or mitosis, don't hesitate to ask, and I will see you in the next video.